Welcome back to Chapter 3, Amino Acids, Peptides and Proteins. In this lecture, which is the part 3 of this chapter, I will introduce you to peptides and proteins. We'll see as to how peptides are made from amino acids and how peptides differ from proteins. In addition, I will also tell you as to how one can purify recombinant proteins. What are the different ways of purification, and this will help you in laboratory purification of proteins. How are peptides formed? Amino acids are linked together to form peptides. The peptide bond, as it is called, is a condensation reaction between the amino group, which is the N-terminus of one amino acid, and the carboxy terminus or the C-terminus of another amino acid. Peptides are short polymers of amino acids, while proteins are longer polymers. We'll talk about this a little more as we go along. Peptides can range from 2 to 40 amino acids. So if you combine two amino acids, it is a peptide because it has a peptide bond. Anything above 40 amino acids, it'll be considered as oligopeptide, and it'll come under the realm of a protein. So, how are peptides made? Let us consider three amino acids. This one, alanine, right? It has a methyl substituent as the R group. What is this? It's a serine, right? It has a hydroxy substitute attached to the beta carbon. This is the alpha carbon. What about this? Tryptophan, right? A very important aromatic amino acid, which is fluorescent. Now, can you make peptides from all three of these amino acids? Let us keep this sequence the same. So, if you combine alanine with serine, you get alanyl serine, and there is a peptide bond that is being formed. And how is it formed? And it is formed by a condensation reaction. This oxygen and the two hydrogens from the amine is removed as water, and you get a condensation reaction. Like I said in the previous slide, it is the peptide bond is formed by combining the amino terminus, the NH2 of the amino terminus of serine, and the carboxylate of the carboxy terminus of alanine, and you get a peptide bond. This amide bond, CONH bond, is called as a peptide bond. Again, if you combine the carboxy terminus of this dipeptide with a tryptophan, you get a tripeptide, right? Now, if you look at this tripeptide, can you tell me as to how many ionizing groups there are in this tripeptide? Let's walk through this molecule, right? The amino terminus NH2 is an ionizable group, right? It has a pKa. Remember, the pKa of this group is close to 9.6. What about methyl? It's not an ionizable group, right? What about OH, CH2OH in serine? Not ionizable, right? And what about tryptophan? This NH is also not ionizable within the pKa range. What about the carboxylate? It is ionizable, right? So it has a pKa of close to 2.5 or 2 between 1.9 and 2.5. So there are two ionizable groups in this peptide. Let us consider a different peptide in this slide. Now, what I want you to take away from this slide is that peptide ends are not the same. The amino terminal end has serine in this peptide, whereas the carboxy terminal end has a leucine. So you cannot swap the amino terminal with the carboxy terminal because the sequence of the peptide matters. By sequence, I mean the arrangement of these amino acids in this peptide. The first amino acid which is serine, followed by glycine, followed by tyrosine, followed by alanine, followed by leucine. And this peptide has a signature, and that signature is the sequence. If you change the sequence, the signature of this peptide changes. Now, which amino acid side chain has an ionizable functional group? And it is tyrosine, right? Tyrosine is the only amino acid in this peptide that has an ionizable functional group, and that is this hydroxy. This is because phenyl hydroxy is much more acidic than the alkyl hydroxy. This has a pKa of close to 10, right? Now, if you were to name this, you have to start with the amino terminal. So, if you were to name the three-letter word, it's ser, gly, tyre, ala, lu. If you were to name the one-letter abbreviation, it'll be S, G, Y, A, L. So this slide is just reiterating the fact that I told you in the previous one, right? The naming of that peptide, you could name it as seryl, glycyl, tyrosyl, alanyl, leucine, which is extremely long if you were to name this peptide. And if the peptide were to be more than 10 amino acids, it's going to be extremely difficult. And hence, 3-letter abbreviation is used for small peptides. And for proteins, 1-letter abbreviation is used, because proteins mostly have more than 40 or 50 amino acids and about 900 to 1,000 amino acids for larger proteins, right? A big point, peptides and proteins also ionize like amino acids. Peptides and proteins undergo the same ionization processes as free amino acid. So if you were to take a small tripeptide, that peptide can also have a titration curve, just like an amino acid, right? And it can have various different pKa's and a pi, an isoelectric point. A decapeptide, 10 amino acids, will only have one free carboxy terminus and one free N-terminus or amino terminus, because all the other amino terminuses and carboxy terminus are joined together by peptide bonds. Ionization properties of side chains are not affected much by the peptide bond. Ionization properties of both termini and the side chains are more affected by the environment and the structure. To this point, as to why ionization property of side chains are not affected by the peptide bond is because there is not much of any electronic effects that a peptide bond contributes, and hence the ionization properties are not much affected. Ionization properties of both termini and the side chains are more affected by environment and structure. This goes back to the original discussion as to why hydrophobic amino acids would like to be on the interior of a protein and hydrophilic amino acids would like to be on the exterior. And again, if you remember chapter 2, the hydrophobic effect, that would be to increase entropy as much as possible for the system, right? And in this case, if a hydrophilic amino acid happened to be in the interior of the protein tries to decrease the pKa of that hydrophilic side chain, and so that it loses a charge, become neutral, and be happy in a hydrophobic environment. We'll talk more about this later on. The next question is, where do you draw the line? How do you distinguish between a peptide and a protein, right? It all comes down to size. Sizes of peptides and proteins vary. Peptides are short polymers of amino acids, while proteins are longer polymers. Peptides, like I said before, can range from 2 to about 40 amino acids. Proteins are typically larger, from 100 to 1,000 amino acids in length. Some very large proteins can be greater than 5,000 amino acids in length. Protein size is often measured in number of amino acids as well as molecular weight. So the two important phenomena. One is the molecular weight and the other one is the amino acid, number of amino acids. These two determine the size of a protein. Another thing that determines a protein and makes a protein unique is the sequence of amino acids. The amino acid sequence of a protein is known as the primary sequence or primary structure. Arrangement of amino acids in standard structural patterns is the secondary structure. The overall fold or structure is called tertiary structure. And finally, the oligomerization state is usually referred to as a quaternary structure. So to explain it to you in little detail, the primary structure could be just the sequence of amino acids. Remember, the sequence in this case starts from proline and ends with valine, right? Now, if the sequence starts with valine and ends with proline, it's a completely different protein. Remember, the sequence matters. The secondary structure is some sort of a unique secondary element of the primary sequence. Now, you may have heard about alpha helices, beta sheets, gamma turns, and beta turns. These are the secondary structural elements. And when the primary structure or primary sequence of a protein takes the shape of a secondary element, it is the secondary structure. And when you combine all the secondary structural elements together, you get a tertiary structure or a polypeptide chain. And this is when a protein takes shape. Now, a quaternary structure is when a single protein dimerizes or trimerizes or tetramerizes and forms various different subunits. And this is called as a quaternary structure of a protein. In this section, we will learn as to how we can work with recombinant proteins. Biochemists' understanding of protein structure and function is derived from the study of many individual proteins. To study a protein in detail, a biochemist must be able to separate it from other proteins in pure form and must have the techniques to determine its properties. The necessary methods come from what is called as protein chemistry, which is a discipline as old as biochemistry itself and one that retains a central position in biochemical research. So, that is one of the very reasons why protein extraction and purification is considered to be one of the top experiments in biochemistry lab. Now, let us see as to how we can work with proteins. Proteins are usually isolated from cells. What kind of cells? Could be a bacterial cell, yeast cell, could be a tissue, or cultured mammalian cells. Now, we are more interested in isolating proteins from bacteria. It's a much more inexpensive way of isolating proteins. And genetic engineering has given rise to synthetic biology, which is being used by various biochemists to engineer recombinant proteins. The first step is usually isolating protein components from other classes of biomolecules like nucleic acids, lipids, etc. because a cell has various different components in addition to proteins, right? Now, the reason why we are using synthetic biology is to engineer cells in such a way that a bacterial cell can express protein, overexpress protein of our own choice. So, a majority of proteins that this growing bacteria expresses is the protein of our choice. Next comes a series of purification steps, collectively known as fractionation. Some proteins require very careful handling to retain structure and activity as well. Protein purification methods exploit the fact that different proteins have a different primary sequence and have different chemical properties. So what different chemical properties could different proteins have, right? One of them is solubility. Other is size, because different amino acid sequences and results in different molecular weights, right? Charges, right? Remember isoelectric point of an amino acid, of a peptide, and a protein also has a pi. So charge varies, right? Does it have a predominant positive charge, or does it have a predominant negative charge? And binding capacities can also be exploited. A protein can bind to various different surfaces. It can bind to various other proteins and to various different resins as well. Now remember that nothing is perfect. And when you do an experiment, there is always some loss of material. But it doesn't matter, because you are doing this experiment and this is just for learning purpose. These are the steps involved in purifying a protein. First, the cells, could be bacterial cells or yeast cells, needs to be broken down to obtain a crude cell extract or a cell lysate. The reason is because proteins are expressed within the cell, and you need to break the cell membrane of a bacteria to let proteins out. Fractionation of various proteins are based on size, charge, or even solubility. Salting out is one experiment that uses the solubility of various different proteins. And in this experiment, the solubility of proteins is lowered in the presence of some salts like ammonium sulfate. The addition of this salt in the right amount can selectively precipitate some proteins while others remain in solution. And your experiment would be to find out this concentration of ammonium sulfate to salt out your protein that you want to purify. Next comes a separation method called dialysis. And dialysis is a method by which you separate proteins from small solutes. The way you do this experiment is you take a bag, a small plastic-like bag, which is not plastic. It's made out of a membrane with small pores. And these small pores can retain proteins within them, but allows the passage of small solutes like salts, and in this case, ammonium sulfate. One of the reasons why you do this dialysis experiment is to remove excess ammonium sulfate that you may have used to salt out the protein of your choice. Once you dialyze the protein of your choice and remove all the excess salts and other small solutes, you perform what is called as a column chromatography. You may have heard this word before in Argo, right? Argo 1 or Argo 2, or you may have even done this experiment. A column chromatography is nothing but uses a column with a static phase and a mobile phase, right? And in this case, you do not use silica gel, but use various different resins to separate proteins by charge, size, binding affinity, and other properties. We'll talk about this in detail. Chromatography is the most common method of purification. Right? Protein of interest is soluble and passed over a porous solid gel matrix that separate molecules by some property. Right? The basic principle is shown here. Right? You have a column, and as you can see, a reservoir, and in this case, it will be filled with buffer. Right? There is a stationary phase, or a static phase, as I would call it, which is made up of a solid porous matrix. And what is this matrix? That depends on what kind of chromatography you're performing. Right? And there is a porous support at the end, and you can pass the protein, you can put the protein sample, load the protein sample here, and then pass your buffer through. And as the buffer goes and passes through, you separate proteins by either size or charge or binding affinity of the protein to this specific matrix, right? Now, there are three major kinds of protein purification techniques. One is called as a gel filtration or size exclusion, which makes use of the size of the protein. And based on the size, proteins are separated. Ion exchange. Ion exchange is based on the charge of the protein. Proteins are separated based on their charges. Affinity chromatography is based on the binding affinity of proteins to a specific solid support. Size exclusion chromatography, also called as gel filtration chromatography, separates proteins according to size. In this method, large proteins emerge from the column sooner than the smaller ones, a somewhat counterintuitive result. You might expect smaller proteins to elute first and then the larger proteins. This doesn't happen. And the large ones comes out first because the solid phase, the matrix that this gel is made up of, consists of cross-linked polymer beads with engineered pores or cavities of a particular size. So large proteins cannot enter the cavities and take a shorter path through the column around the beads, as you can see from this diagram here. Small proteins enter the cavities and are slowed by their unusual path and hence take a longer route and elute later. This slide just reiterates what I told you in the previous one, but it gives you a much more refined explanation, right? And you have these beads that are the blue colored ones, and the red colored large protein molecules cannot enter these beads, whereas the pink colored or the magenta colored small proteins can enter them. Larger proteins just circumvent these beads and take a shorter path and come out, right? Because large particles are excluded. They have less volume to traverse and elute sooner. Small particles can enter gel and have more volume to traverse, elute later. So if you look at the profile, right? Elution profile, this is the larger protein and the magenta is the smaller protein. Next is a technique called ion exchange chromatography. This technique exploits differences in the sign and magnitude of the net electric charge of proteins at a given pH. So basically, you separate proteins based on their charges. The column matrix, right? The column matrix is the one that the column is made up of, which is the stationary phase. It is a synthetic polymer or resin consisting bound charge groups. Those with bound anionic groups are called as cation exchangers, and those with bound cationic groups are called anion exchangers. So let me make it more simple. If this bound resin has a positive charge, right, it is called as an anion exchanger. If this bound resin has a negative charge, then it is called as a cation exchanger. The affinity of each protein for the charged groups on the column is affected by the pH and the concentration of competing free salt ions in the buffer or the solution. Now the reason is because when you load the proteins and when you load the buffer, The buffer also should have some sodium chloride or some salt solution. Sodium chloride is the commonly used salt in ion exchange chromatography because it has sodium ion, which is positively charged, chloride ion, which is negatively charged, and these will compete with either a cation or an anion exchange column. And it will also compete with the proteins, thereby resulting in elution of them. In cation exchange chromatography, remember, cation exchange is when the resin has a negative charge, right? These gray balls are all the resin, right? This is a cation exchange. And in cation exchange chromatography, the solid matrix has a negative charge. In the mobile phase, proteins with a net positive charge migrate through the matrix slowly than those proteins with a negative charge, because the negatively charged proteins will repel these negative charges and they'll migrate much faster as compared to positively charged proteins, which get attracted to the negatively charged resin. In ion exchange columns, the expansion of the protein band in the mobile phase is caused by separation of proteins with different properties and by diffusional spreading. What do I mean by that? Shown here is a chromatogram from an ion exchange chromatography column, right? And in this chromatogram, the way you read this chromatogram is by looking at the y-axis and x-axis. On the y-axis is the absorbance and x-axis is the fraction number. What this means is that while collecting, you're collecting in various different fraction tubes. And it's a number of the tubes that's being shown here, right? There's another axis, and that is a sodium chloride concentration. Remember, I said sodium chloride in the previous slide. It's because only if you increase the concentration of sodium chloride can you elute the protein from an ion exchange column. Now, how do you monitor the elution of this protein? That's by absorbance. Why absorbance? Because proteins have aromatic amino acids, especially tryptophan and tyrosines, which can absorb at 280 nanometers. And that's where you observe this, right? Now, what kind of an ion exchange chromatography is this? I don't know. It could be either an anion exchange or a cation exchange, right? Now, let's assume it's a cation exchange chromatography because that's what we saw in the previous slide, right? In a cation exchange chromatography, the beads or the resin is negatively charged, so it retains positively charged proteins. So what is the first protein that comes out? This is more likely a negatively charged protein, right? Now, negatively charged protein just flows through the column. It doesn't even stop anywhere. And it doesn't even require sodium chloride to push it out. By the way, sodium chloride, what it does is, in a cation exchange chromatography, the sodium ions compete with the proteins. As you increase the concentration of sodium chloride, sodium ions slowly replaces all the positively charged proteins. And the more positively charged proteins get eluted last, right? The less positively charged protein elutes somewhere in the middle. And the negatively charged proteins does not even need sodium chloride to elute it. Now, the elution of this protein also depends on another factor, right? And that factor is the length of the column. As the length of the column increases, the resolution of two types of protein with different net charges generally improves. For example, if in this case, protein 5 and 6 are a little closer to each other, if you were to increase the column length, you may be able to separate these two proteins much better than as you see here in this chromatogram. So, from your perspective, what is it that you need to know, right? Especially with regard to this course. When I talk about 3 different methods of protein purification, size-exclusion, ion exchange, or affinity purification, I want you to remember and get familiarized with how to interpret a chromatogram. Now, I explain here the example of ion exchange chromatography, right? How do you interpret this? Now, I need you to familiarize yourselves with how to interpret a size-exclusion chromatography or an affinity chromatography, right? So that's something that I expect from you. Affinity chromatography is based on binding affinity of a protein to the stationary phase in the column. The stationary phase in this case are called beads. The beads in the column have a covalently attached chemical group called a ligand, a group or molecule that binds to a macromolecule like protein. When a protein mixture is added to the column, any protein with affinity for this ligand binds to the beads. So this protein is essentially retained in the column, and its migration to the matrix is retarded. And then later on, a solution of a ligand in the buffer is added to the column so as to decrease the affinity of the proteins to the ligands that are attached to the beads. So what happens is, as the concentration of the ligand in the solution increases, the protein binds to the ligand in the buffer and detaches itself from the stationary phase. And hence, these proteins are eluted. Now, how does this help in purification? Is because in the first step, when the protein is added, only the proteins that have an affinity to the ligand will get stuck in the column. The rest of the proteins would have got eluted in the first place. So hence, you see a separation. You see 3 and 4 are impure proteins, whereas 7 would be your pure protein. There are other separation methods in addition to these 3 column chromatography methods. One of the classic methods is an electrophoresis method. Now, electrophoresis is a method of separating proteins by applying an electric field. Polyacrylamide gel electrophoresis, or PAGE, is a common method for characterizing protein size and properties. Proteins, when unstructured, will migrate in a polyacrylamide gel according to their size. Typically, a combination of detergent, usually sodium dodecyl sulfate, reducing agents, and heat is used to unfold proteins for polyacrylamide gel electrophoresis. I'll talk to you more about this in the next part.